So I'm Ryan Muldoon. I'm co-founder and CEO of RNA Connect, and I'm really grateful to have the opportunity to introduce the company to you today. We founded RNA Connect 3 months ago with a mission to provide better tools for RNA researchers. And specifically, what we have are a suite of enzymes that are foundational technologies for interrogating RNA. We think the existing suite of enzymes that everyone uses are, in a word, terrible. And so we're working to provide better ones for the community. We think what that will enable is whole new levels of innovation, because when you have better tools, you're able to create bigger, better, faster things. So for us, our lead compound, our lead enzyme, is a novel and highly differentiated reverse transcriptase. We think what this will allow us to do is have dramatically better viral pandemic surveillance. It'll enable us to do better target identification and biomarkers for cancer and neurodegenerative diseases. And importantly, it's going to provide dramatically better applications in RNA sequencing. So what is reverse transcriptase? It is an enzyme that latches onto single-stranded RNA, and like a zipper, it creates a complementary strand of DNA, which can then be amplified for your PCR COVID tests. It can be sequenced by the DNA sequencing machines from Illumina and PacBio and others. And there are many reverse transcriptases that are on the market already. The trouble with them is that they are from the early 1970s and were in no way designed to do what we're using them for today. So we have all sorts of workarounds to overcome their shortcomings, which are specifically related to processivity. What that means is it's how long the enzyme will continue copying the RNA template before it falls off. And the existing RTs will go 100 to 200 bases before they simply fall off the template. That's a problem because most RNAs are 2,000 to 30,000 bases long. So you end up with these little snippets of information, but you're never able to see the whole RNA. This is confounded also by the fact that RNAs are highly structured. They have all sorts of loops and stems and secondary folds in them, and each time the existing RTs encounter one of those, again, they fall off the template. So there's all sorts of information in those secondary structures that are essentially invisible to us right now because the RTs can't get through them. So what is our solution? Our solution is Marathon RT, Marathon Reverse Transcriptase, which is a novel RT that was discovered right here at the Yale Pile Lab. It was optimized and the structure was solved here. And when it was initially tested, we looked at, okay, well, how processive is it? Can it read through an RNA transcript? And we gave it the Hep C genome, 10,000 bases long, and it read the entire thing no problem. This was fairly astounding when we said, okay, well, what else can it do? And I love the expression for anyone that's at least my age. We gave it what Dr. Pyle likes to refer to as stupid pet tricks. And we inserted all sorts of little RNA structures, secondary structures into it and said, okay, well, can it read through? And let's compare it to the market-leading reverse transcriptase, which is superscript 4. And what we saw was superscript 4 does what it does. Every time it hit one of these inserted structures, it fell off the template. But Marathon RT read all the way through. It unwound all of those structures and gave us the full cDNAs for those RNA transcripts. So this is a vastly improved reverse transcriptase that will enable all sorts of new applications, particularly related to RNA sequencing. Imagine now we can get better single-cell sequencing. We can read all of the RNAs within a single cell. So what we're doing is we're packaging this enzyme. It's already fully optimized. It's ready to go. We're putting it with the other reagents into kits, and in the next 6 months, we'll begin commercializing them. So this will be for all sorts of sequencing applications, as well as for RT-PCR applications. We'll have products in the market in the coming months. Ultimately, this will enable new diagnostic applications, companion diagnostics for cancer, for neurodegenerative diseases. So we'll be looking to start those programs as soon as we can get the company off and running. The company was founded And we have a scientific advisory board that are RNA researchers. These are leaders in the field, people that many of you, I'm sure, know, that are helping us then shape, okay, so what are we going to do with this enzyme as well as the rest of the enzymes that are in the portfolio to come. So I thank you for your time, and we look forward to enabling all of those that are in the RNA research community with our new tools.